This is a presentation on the filaments in the cytoskeleton and their function. The cytoskeleton is a complex network of protein filaments within cells. They play 5 key roles in cells, providing structure for the cell, organization of the internal components of a given cell, mobility, or helping a cell move around if it has that function, transport of vesicles and organelles within a cell, and they also assist with division of cells. There are 3 types of filaments in the cytoskeleton: actin filaments, also known as microfilaments, intermediate filaments, and microtubules. Actin filaments are polar chains of actin. Monomers attach at the positive end and detach from the negative end. They can use this to move around in a process called treadmilling. Their structure is a flexible coiled chain, and their roles in the cell are involved in structural support, division of the cell, movement, one particular example being cell crawling, and intracellular transport. Cell crawling can be seen in that diagram to the side. Basically, an ARP complex and formins produce a protrusion from the cell. Integrins then attach that cell to the new place, and myosin II provides traction for the cell. And it just continues using that process of sending out a piece and then attaching and coming and then moving the body over just a little bit to move around. And most of these actin filaments can be found concentrated right under the membrane. All right, myosin. Myosin is a binding protein to actin. ATP detaches myosin from actin. This is important because it gets into an effect we're going to talk about later. Myosin 1 is the basic structure. It has a head and a tail. The head binds to actin and moves by attaching and detaching in certain patterns. Myosin 2 has a more complex structure and functions in a more complex mechanism. It's used in muscle cells for contraction, and basically each has a dimer with 2 globular ATPase heads and a tail. These in turn form myosin filaments, which in muscle cells some face one way and some face the other way. Myosin filaments pull actin towards the center gradually, and the contraction is mediated by calcium and accessory proteins. So, in muscles, skeletal muscles in animals are usually large and multinucleated, formed of myofibrils, which in turn consist of repeating sarcomeres. Sarcomeres have actin and myosin filaments in that grid pattern, which you can see in that picture off to the side. And this makes the power stroke, which I'll talk about here in just a second, pull the actin towards the center, shortening the specific sarcomere, which in turn shortens the muscle that it's a part of. The thick filaments in the sarcomere are myosin II, and the thin filaments are actin. So the mechanism by which myosin is moving on that actin, the calcium ion exposes the myosin binding sites on actin. ATP provides the energy necessary to undergo that power stroke, and without additional ATP, the myosin can't unbind, which because of conditions affiliated with death leads to rigor mortis after death, because there's not ATP stopping the muscles from contracting and stopping that binding of myosin in the muscles. All right, intermediate filaments. These are usually described as rope-like. Uh, they are responsible for dealing with the mechanical stress placed on cells and are the toughest and most durable filaments. They're the only non-polar filaments, and they have dimers facing in opposite directions. Both ends assemble at the same time, and if they're dysfunctional, cells can become misshapen and damaged. Accessory proteins reinforce them by linking them into bundles and connecting them to other filaments. So these are basically responsible for providing a lot of the structure of cells and making sure that things don't break down and have the strength to actually deal with external stresses. All right, microtubules. Uh, these are important for organizational and movement purposes. They're long and hollow. They rapidly disassemble and reassemble, engaging in dynamic instability, which is where they shorten or lengthen quite rapidly and relatively spontaneously, unless other proteins are keeping it stable. They're polar, which assists with their directional transport. In the centrosome, animal cells have a centrosome, and this consists of two centrioles with microtubules radiating out from nucleation sites around them. What the centrioles do is still quite mysterious, uh, and they're the main organizational center for microtubules in animal cells. Other proteins can use it like a train rail. One example of this is myosin walking across it, uh, as you can see in the image off to the side there. And they're also used in cilia and flagella. Just as a side note, videos on how this works are really interesting to watch because the interactions are quite fascinating. All right, that's all. Thanks for watching.